Okay. Hello, I'm Dr. James Camp and from Lee College in Baytown, Texas, and this is my overview of nerve signal conduction. In today's lecture, we're going to delineate the functional regions of a neuron, give an overview of the events of nerve signal propagation, explain axon diameter and myelination, and explain the role of myelin in something called saltatory conduction. Okay. So first, let's talk about the parts of a neuron, which you should by now know. Um, we have here the soma, or cell body, okay? That's roughly this part of the neuron. We have back here the dendrites, okay? Which are each of these little tree branch structures going off into space here. We have a little transitional zone here called the axon hillock, which will be important in this discussion, be made important in this discussion in just a minute. We have the axon. Okay. We have the axon, okay, which is this whole long bit of neuron that stretches, in some cases, just a short way from its cell body, and in some cases, it can go for over a meter in length. We have telodendria here at the far end of the cell. Again, sort of a tree branch-like structure. Dendros means tree, so it's a tree structure, but this is telodendria, the tree at the end, is what that means. And then at the absolute end of each telodendrion, we have these little balls called axon terminals. Or some books call them synaptic. Whoa, I don't know why my pen is out of control here today. Let's try this again. Some books call these synaptic knobs. Okay. So we've got all sorts of parts to the cell here. We also have these little kolache rolls here. Called Schwann cells, or some books now refer to them as neurolimacytes because they hug the neurolima, the conductive portion of the cell membrane. And then here in between, in the little spaces between the neurolimacytes, between the Schwann cells, we have nodes. little spaces where a little bit of cell membrane is exposed. Okay, now we're going to talk about the regions of a neuron again, but this time from a different point of view. This area here is all functionally responsible for receiving incoming signals from other neurons. And so it is called the receptive segment. There is then a section here that is responsible for initiating action potentials, and so it is called the initial segment. That's a little confusing to some people because initial, we usually think that's the very first step in something. It's not in this case. The reception is the first step, but this is initiating. The reception is receiving other people's nerve signals, other cells' nerve signals. This is where we initiate our own unique nerve signal. And all of this is the conductive segment. It's the propagation. This is where an action potential travels quickly down the cell and up its various telodendria. And then finally, at the very end, all of these synaptic knobs make up a transmissive segment. And we're about to talk about what each of those do, but first, no, we are. Okay, so the receptive segment's job is to listen for incoming signals, to receive incoming signals. The purpose of the initial segment is to sum up. Okay, you learned sometime in elementary school that adding things up is called summing them, so we have a process called summation. The initial segment sums up incoming signals until, I should say sums up, and listens. The receptive segment listens for incoming signals. The initial segment sums up incoming signals until a threshold is reached to start an action potential. The conductive segment propagates, which means passes along, an action potential. So the action potential propagates from one point on the cell to another, to another, to another. And then finally, the transmissive segment, guess what? Transmits the signal to the next cell in line. Okay. Before I get to my final slide summing this process up, or explaining this process in a little more detail, we have one more terminology to introduce, and that is this thing called saltatory conduction. two things affect the speed at which a nerve signal is propagated down an axon. One is the width of the axon. Just as kind of a wider, thicker copper wire is going to carry electricity faster, a wider, thicker axon is going to carry signals faster. So if I look here, a neuron that looks like this is going to be relatively slow. A neuron that looks like this is going to be relatively fast. But the other thing that affects it even more, much more than the narrowness or wideness of the axon, is whether or not it has myelin, whether or not it has these neurolimocytes providing, as you recall, they provide a myelin sheath, several layers of cell membrane here on either side of the neuron that insulate it. Nothing is going to happen at the cell membrane where it is covered with myelin. So what happens instead is that the action potential happens here at the axon hillock, and then it appears to jump to the first node. Then it jumps to the second node, jumps to the third node, and etc. And that jumping process happens very, very quickly. It happens at the speed of an electric field, which is much, much faster than the speed of neuron electrical signals. So this dramatically speeds up the conduction of an action potential down the neuron. We call this process saltatory conduction because the word for jump in Latin is salta. It is in Spanish as well, so if you're a Spanish speaker, you've got a leg up on the rest of us here. Okay. Finally, we can wrap this all up with 5 phases of a neural signal. Okay, so once again, we had our segments. Initial, oh no, see, I did it this time. Put initial first just because it has that name. Our receptive segment carries out the first 2 major phases of the neuron signal. Then we get to our initial segment, then our conductive segment, and finally our transmissive segment does its job down here. So what are the 5 steps of a neural signal? Well, first off, I have to just explain how I see a neuron, which is a little bit different perhaps than some other people, perhaps than your textbook. And that is, I see this neuron as having a upstream end and a downstream end, because information is always passing along this neuron in one direction. And that, just as water in a river travels from upstream to downstream, information in a neuron travels from a sort of upstream side to downstream. So we start all around here with upstream neurotransmission. Okay, we have to get a signal from an upstream neuron. Maybe we've got another neuron coming in here, okay, that passes the signal along. We've got to have a neurotransmission event from the upstream neuron. Then the potential that is generated by that neurotransmission event creates a local or graded potential on the receptive segment that travels along the receptive segment. Local potentials are called local because they don't travel very far. Okay, if you say eat local, you mean you want to get food that's grown within a few dozen miles of where you live. Similarly, a local potential only travels a few micrometers along the cell. It's certainly not going to make it all the way to the downstream end of the cell. They're called graded potentials because just as you will get an A, B, C, or D in this class, a potential might be plus 10 millivolts or plus 5 millivolts or minus 5 millivolts or minus 15 millivolts. You know, we don't know. There aren't necessarily universal numbers for the various graded potential grades, but they do come in different grades of high or low potential change. If you're getting a little thrown off by all this talk of potentials, don't worry, that's in my next lecture. Okay. The action potential, so first off, at the axon hillock now, we sum up. So summation of those graded potentials takes place. So let's say you get a plus 10 and a minus 5 and a plus 5. That's going to sum up to a plus 10. And you get another plus 10, and now it's a plus 20. Okay. They sum up until a threshold potential is reached. So what's a threshold? Well, if you know in real life, the threshold is the little thing under your door that men are supposed to romantically carry their wives across after they're married, that sort of thing. Anyway, a threshold is something that you cross to get from one place to another. And in science, thresholds are generally looked at as when you make it to a certain value and a sort of magical change happens. Obviously not really magical, it's scientific, but a apparently magical change happens and you see a different behavior. So instead of walking up the potential curve gradually, suddenly it shoots up to a full action potential. Okay, so then we have an action potential that travels all the way down our neuron to the various telodendria. These are called action potentials because they are not local or graded. They are all or nothing signals, so they're not graded. It's always a certain potential or it's nothing at all. And they very rapidly travel down the axon all the way to the terminal. So they are not local, they travel all the way to the end of the cell. Then finally, here at the end, we have downstream neurotransmission occurring as our cells release their neurotransmitters on to other cells that might be waiting for them here and here and here. Okay, so those are the 5 basic phases we're going to go back through. In my next lecture, I'm going to talk about what potentials are. Okay, the physics, you get a free physics lesson with your biology subscription. And then in the lecture after that, I'm going to talk about local potentials, how they sum up, and what an action potential is.
Did you find my phone?
And then finally, in our last lecture, I'll talk about the neurotransmission event and how that carries a signal from one cell to another across a synapse. All right. Happy studying, and I'll see you in the next lecture.